So hopefully today I'll be able to show you how you can use a multiscale imaging approach to tackle a very complicated catalyst here. So you'll see this slide later on in the presentation also. But hopefully I'll be highlighting the image there on the right here, a GPF. So unfortunately I couldn't bring one on the plane to show you guys in person, but here's a nice one that way. So I'll give you a brief introduction into GPFs. We've had some fantastic talks about those so far. And then going into some of the results we've had about changes in the activity and how we use a multi-scale imaging approach to understand this system further. So what is a GPF? So essentially it's a three-way catalyst, so a palladium, rhodium, silver-zirconia, aluminum-based catalyst, which is converts CO, maximum number of hydrocarbons, which you guys have heard earlier. But GPF is slightly different, where adjacent channels have been plugged here, so that actually captures particular matter. So this is soot and ash, which is formed from inorganic, combustion of inorganic materials, such as from additives, such as engine lubrication. So as emission standards get more stricter, in order to adhere to these, you need to improve the GPF technology. Oh, thank you. Okay. Clearly, I'm more confident about this. There we go. Yeah, so particularly the particular matter. So the theoretical deposits on the GPF surface interacting with catalyst wash flow. So over time, as you age the catalyst, the condition change, experiencing high temperatures over its use. The sort of particular matter in the catalyst interaction varies, especially in the ash. So it's normally the soot will oxidize off, but the ash being an incombustible inorganic stays on the surface. So it's important to understand how the elements in the ash actually interact with the catalyst. Does it change the activity over time? Does the distribution of the elements vary upon these aging procedures? And those are information about the catalyst, such as changes in the morphology. So we did some initial, this is published, but we've done some operando XF studies on this catalyst. Won't go over too much into the XF details, but the main crux of it for this presentation is that we saw changes in the CO, NO, and drop in light-off temperatures. So we took two catalysts, one from GPF, one from a GPF which had pre-ash loaded, and what we found with the pre-ash loaded GPF is that we saw an increase in the light-off temperatures being present there. So we're seeing this change with ash being present in the system. So now we are looked at using a multi-scale imaging approach to understand what is actually going on, what speciations are actually affecting the system. So I'll be going over some of these techniques right now. So to reiterate the point, we've gone from an actual GPF and retained the spatial resolution, so looked at the sample in the same region of interest using multiple techniques. So we're talking lab-based techniques, such as Xrmct, lab-based techniques listed Xrmct, EPMA, FibSEM, which might be more aware of. I'm going into a bit more sophisticated techniques, including nanoprobe XRF. So this was done at the I-14 beamline at Diamond Light Source in the UK, and doing algorithm corrected stem microscopy done at the Xrmct also in the UK. So we're again taking the same sample, going across all these all these different techniques of high field of view, low resolution to low field of view, high resolution to understand the sample and get a bigger information about the system going on. So today I'm going to be briefly going over the lab-based techniques so you guys get an appreciation of what's going on, but mainly focusing on the ones here in blue and the ones here in yellow going on ahead. So a bit about the samples before I go into the results. So I'm going to be showing you samples which again moved from two GPFs. One which has no ash deposited and one ash loaded deposited. They're taken from the inlet of the GPF, so these samples which are showing, and we've looked at the same region of interest, so we're retaining spatial resolution. I'm going to be showing you samples which have been aged in two different methods, so aged in a reactor, and both originally aging, and aged on an actual engine. So I'll be showing you how differences in the Bayesian procedures that you perform actually affect the distribution of the elements present in the system. So let's get cracking into some results. So the first one I'm going to show you is Xrmct. So this is a tomography absorption study. So basically gives you a nice overview of the 3D structure of the GPF and tells us the distribution of the catalyst wash hole, which is a bright color here, and the room relative to quarter support. So the main information we actually found from this is that with the pre-ash deposited GPF, the one on the right here, it's quite hard to see here, but in the top right corner, the ash is deposited in this channel, but the actual catalyst wash cup is in the adjacent sample channel, which isn't the case in the non-ash sample. So we believe that this is actually due to the manufacturing of the GPFs. So as the adhere the catalyst wash cup onto the walls, they actually vacuum dried, so we believe that it's actually been pulled through to the adjacent channel. So this gives us great information. It's not a lot of what's going on with the system, but again, it doesn't tell us elemental information what these different species are doing. So we use another technique. So this is electron probe analysis. So we took a region of interest, what we found from the XRM, and we use this study, which is essentially elemental mapping using wave dispersion detector. So what I'm showing you here is zinc and palladium. So zinc and calcium are two of the main components that we found from the ash itself. So what we're seeing upon engine aging is that the zinc component is actually moving into the GPF wash cup region, which coincides with where the palladium is also, which is great. So we're seeing some variation in some of the ash components. But the resolution that we're seeing is quite low, so we can't truly understand if there is actual co-location between palladium and zinc at this stage. So we wanted to take it to something with a bit more higher resolution. So this was the nanoprobe beamline nanoprobe Xrmct mapping, which I'll be showing you there. So I did go from a lab to a synchrotron. We used FibSEM, so this was our sample prep way. Again, SEM, quite a lot you might be aware of the study, so we can get understanding about from the micrograph. So this initially showed us that our hypothesis of the pull of the GPF logical to the adjacent layer. There's more information about that. And also with it, with the Fib side, so called this iron beam, we could actually extract a region of interest from this GPF. So here's some regions of interest here. Again, with SEM, we can use EDS, but this is relatively time consuming and doesn't tell us any information about the oxidation state of the elements present, which I'll hopefully be showing you now that you can do using nanoprobe, the beamline, which I'll be showing you later. So, as I was saying, we found GPF, found a region of interest, extracted it using a focused iron beam, which is a gallium iron gun. You extract it, put it on a copper grid, and stuck it onto the beamline. So, here's a nice picture of the beamline. So, this is, again, in the UK, kind of light source. So, if you're not familiar with fluorescence mapping, synchrotron-based fluorescence mapping, it's pretty good. So, the beamline here at I-14 has a relatively high resolution of 50 millimeters. You can actually tune the photon, so you can actually maximize the fluorescence output. So you can see a plethora of fluorescent species present, and also you can couple this with other techniques. So you do XRD, differential phase contrast imaging, and spatially resolve the same. So this tells us about spatial state. The latter I'm going to be focusing you on more on how this can help us understand the GPF system. This is particularly important for the zinc. So we've done relatively bulk zinc studies at the B18 beamline at diamond light source. The main take home from this was that we see the sample in yellow here, which is the pre-ash deposited GPF, but on engine aging of the system, the zinc component changes. And we initially did not know what this was, and we initially thought that this was a multi-component system. So this allowed us to use the spatial results, which we'll be showing you later, to try and deconvolute the individual components being present there. So now hopefully nobody's got mine in the audience, but so there's going to be RGB maps showing XRF mapping here. So Palladium here is in red, incoming caps are in green, so essentially what the beam line you see on the beam line. Hopefully the one in the middle you see nothing, so that signifies to us that pre-ash deposition onto the GPF hasn't caused any palladium particle growth. However, upon entering you see growth of palladium species, but especially in a sample containing pre-ash deposition, so you see large palladium species here. So I could be a bit of a charlatan and just showing you images which show my point, but we are capable of doing numerical analysis on these systems. I won't go into too much detail about the actual statistics of it, but the main information that we got was we could highlight the elements present around the palladium species. And what we found was when these ash-loaded systems was on a high it showed us that there was a high amount of zinc present around these palladium species. This is great what we're mapping about these structures. So, so if you map zinc, which is in blue here, and calcium, the other component in the ash in green, what we find is that pre-ash deposition, there's no evidence of it penetrating into the washed-up layer on the left. They're not in the system before pre-ash deposition. But however, after it's on an engine age, you can see, especially the zinc in this one here, is that it's actually migrated into the washed-up layer. When we zoom in onto these regions within high palladium, you see evidence of zinc and calcium signals co-located around these palladium particles. So we see these large palladium particles, evidence of the components from the ash actually being around the system here. So now this Xrmct which I showed you from engine aging. So when you compare it to reactor engine, it's totally different. So hopefully in the middle one you see no blue. So it's signifying to us that the zinc components actually disappeared from the actual aging procedure. So what we found is that the aging procedure doesn't matter how you perform these catalysts. And what we're seeing is that with the, again, just to highlight the point that with the ash-loaded system, we're seeing changes in the catalytic activity. And we're seeing evidence of the zinc component being around these palladium species. So what are the zinc components? So again, this is where we've been showing you spatial results saying this mapping. So we took 5 regions of interest from a pre-ash GPF, not age, and then compared it to the same sample from the same GPF for entry age. What we found was that initially from the pre-ash deposition, the new combination of palladium suggested that this was a combination of oxides, sulfates, and phosphates. The important one was from when we looked at the entry age one, especially the regions where the zinc was found within the wash code, where we actually saw we found zinc veins, which are features which were akin to what we saw in the bulk zane studies which we did at BAT. So this tells us 2 things. It tells us that this is one of the major components in the system for the zinc, and also that it's probably not a multi-component system, it's probably an individual species which you could have identified. So after a lot of literature review and a quick visit to the this museum, actually found it. So this is gannite, so it's a zinc spinel structure which has similar features to what we saw on the from our zinc structure, and we believe that this is actually formed from the zinc component interacting with the aluminous spot and actually reacting and converting it. So we're seeing changes in the excessive pot from the presence of zinc, which is found in the ash upon aging and entering. So so hopefully you guys are still awake and you can see that we have hopefully shown you that we've gone from GPF and taken it through these different large scale techniques and got varying information about the system, but we can take it one step further. So we can take the same sample and do AC stem on it. So any microscopists in the room will know that it's good to have a question when you're doing microscopy. So we have a couple here is that does the XRF correlate with other element studies of EBS? And also does the high resolution of the microscope tell us more information about the palladium species itself? So here's a sample on the right which we've never seen. This is the same sample which we've done with a patchwork micrographs on the left, which are in the microscope. And I'll be focusing here on the region in blue, which I'm highlighting here, so like a nice plane. So when we compare this region, when we do EDS on this region, the one over here on the right, it's showing us multiple things. So firstly, it correlates to what we see in the Xrmct, showing the overlap of zinc and calcium around this palladium species. Secondly, with the higher resolution, we are able to identify more individual palladium components, palladium species being present, which we're going to see here. And again, with the EDS mapping, we can see other elements being present, so getting more information about the environment, including telling us information that there's co-location of zinc and sulfur and calcium with phosphorus. Again, with the microscopy, we can learn to zoom in a little bit more to understand what these actual speciations are. And we believe that these are agglomerations of palladium particles, which can identify with the variation as highlighted as being present here. So, hopefully, I'll be able to show you how you can take a very complicated real-life GPF and use a very multiscale approach to understand the system. In our case, it was an ash molding that's caused affecting catalytic activity, and we found evidence of the migration of components from the ash, namely the zinc, into the catalyst washcoat, interacting with these support material and forming a zinc spinel anode. So again, just to highlight the point, this was actually quite cool. Once you're writing UPH, you kind of realize it. You've actually gone from, I've shown you that we've gone from lab-based techniques to synchrotron to microscope. On the same sample, we retain the region of interest, we'll look at the same region, and hopefully I've been able to show you that the disadvantages of each technique can actually be complemented by the advantages of the other. And that's me done, so I want to thank the people here, mainly the people in red, Xrmct, Tim, probably been in Bray over 4 years, and especially the guys at home, which maybe before it was great. That is me done, so any questions?